so this works. I'm Laura Galloway. This is Biology 3020, Ecology Evolution. Hopefully this is where you want to be. And I am one of the two teachers in this class. The other teacher is Butch Brody. And Butch is just stopping by for a minute now because really you're not going to see him until the second half of the class. So I do the first half of the class, so 2 exams worth, and then Butch takes over and does the second half of the class. The Brittany Sutherland is your TA. A lot of you may know her if you've taken genetics. And Brittany will be here the whole class. So what I want to do today is talk a little bit about course logistics in the beginning. And so if you have any questions, get ready. You're going to ask them. And then we're going to jump right into material. Well, let's see. Okay. There we go. So just a little bit about class. Brittany set up the Piazza site last night, so you should have gotten an email, if you have that enabled, that said that the PowerPoint for today was going to be uploaded onto Colab. So every night before class, I will post a PowerPoint on the course website in Colab, and I will do that by 10 o'clock, let's say, at night, and so you can download the slides there. Also, the syllabus is on Colab, and the syllabus tells you when exams are and what the readings are associated with each class, as well as other sort of class rules, learning goals, etc. Textbook. Oh man, I forgot to bring it. Okay, so I had it out on my desk. So there's this textbook called Evolutionary Analysis, has a nice gorilla on the cover. I'd be holding it had I remembered to bring it. Anyway, there are 2 textbooks in this class. The Evolutionary Analysis will cover all of the evolution lectures. The readings are posted on the syllabus. We use this text for a number of years in this class. Basically, students seem to like it okay. In other words, the course evaluations are not filled with complaints about the textbook. I encourage you to read it. The one thing I will say is that it's a really comprehensive textbook. There's a lot of stuff in it. Sometimes the chapters are long, and we may not cover all the material in the chapter. So here's the important point. I will never test you, Butch will never test you, on material that's in the textbook that we don't talk about in class. So you're looking at the chapter and you're like, oh my God. So just, if we don't talk about it in class, you're not going to get tested on it. Okay. That's sort of basically the text. But do I think the textbook is useful? Yeah, I think the textbook's actually really useful. Okay, exams. There are 4 exams in this class. 3 of them are during the week, the evening, Wednesday 5 to 7 review session. The final one is during the final exam period, but it's not a cumulative final. It's just a 4th exam. The exams, the format is going to be multiple choice, and the way that we work the exams in the class is that I write half the exam questions, or Butch does. Brittany writes half the exam questions. Why do you care about that? You care about that because halfway through the class, I leave and Butch comes, and you think, oh, I have to get used to somebody else's exam question writing style. But actually, Brittany writes half of them, so you get her all the way through the whole class. The other thing is that every week we come up with a set of questions. Half the questions go on to the exam. The other half the questions go to the review session. So if you do the problems for review session, you will have experience with my exam writing style, Butch's exam writing style, Brittany's exam question writing style, right? So when you get questions for the review session, and Brittany will send those out every week, the multiple choice ones, take them seriously, meaning that they reflect real exam questions. So the style, the way they're written, that's what you're going to get at the exams. And I'm thinking, okay, next thing is review sessions. Review sessions are optional. What? You're not having one today, thank you, Brittany. Right, not one today, not enough to do yet. But every other week, except for exam weeks, there's going to be a review session. They're optional. Brittany's a great TA. You come to a review session, you're going to learn more and have to study less. So 1 hour every week versus more hours studying, that's your choice. Okay, so that is the evening review session. Office hours, so every week, or faculty actually have office hours Monday 2 to 3. This is in the syllabus. All the office hours are going to be held in Gilmer 227. And then Brittany has office hours Tuesday, Wednesday 2 to 3, and then Friday 8:30. You may be thinking, 8:30, this is just before class, right? If you want to do anything before class, so this is office hours before class on Friday. You might notice we've structured, even though we hope you're going to come to office hours every week because they're useful, if you don't understand material, you should come. The exams are Wednesday night, so we've usefully structured these so that you can come take advantage of them before exams. But really, come to office hours. I'm there, Gilmer 227, every week, hanging out. Come with questions. If you don't have questions, other people likely have questions, and you may even learn just from listening to their answers. So I really recommend coming to office hours if you're uncertain about any material. Okay, what else do we have to talk about? Piazzas. So as I mentioned earlier, Brittany got that set up last night. Is there anybody who hasn't used Piazza yet? A few people. Okay, so it is a place to ask. It's on the course website. It's a place to ask questions for class. It's like a public forum for questions and answers. Definitely go there to ask logistics about class. Like, for all of you who manage to forget the fact that I've said that you're not responsible for things in the book that I don't cover in class, I guarantee someone will put that question on Piazza, and that question will be answered, and you'll get to read it in text so you know it's absolutely true. It's still true because I said it, but that's where you can get sort of logistic questions like that. And also, people will ask questions about course material on Piazza. If you have a hard question, come to office hours. Don't go to Piazza, because the thing about Piazza is you get an answer that's like this long. That works for an easy question, but not a hard question. But Brittany will check Piazza every morning, every night, so 8 a.m., 8 p.m., and look and see what needs to be answered. You can answer each other's questions too, and I strongly encourage you to do it. If you see a question and you know the answer, just do it. It's good practice for you. If you can answer the questions on Piazza, you can answer the exam questions. And yeah, go for it, Butch.
So we did statistics last year about people who used Piazza. There was a really strong correlation with people who answered questions, other people's questions on Piazza, and they're great at the end of the test, the course. So maybe they were the ones who were going to do well anyway, but it worked.
Or maybe it was like sitting there formulating the answer. How am I going to answer this? Do it. And if you want to answer it and you're not quite right, I mean, you can take a risk. If you're not quite right, somebody else will come along and give what they think is a better answer. So if you answer in Piazza, it doesn't have to be perfect. So just, what?
I will correct anything that's wrong.
Yeah, Brittany will correct anything that's wrong. So don't be shy about answering stuff. Okay, so that's Piazza. Clickers. How many of you brought clickers to class today? Right on, because we will have some clicker questions. So usually, Brittany didn't think you guys were going to bring them. I said absolutely, we'll bring them. Yeah, so usually we'll have a couple clicker questions per day. All right, luckily we did that for class. I think that's it. Okay, so on to first section of the class. See you later. So this is actually totally an aside, but Butch leaving makes me think of this. we're having a faculty job search for an evolutionary ecologist this semester. So we have 6 people coming in to give job interviews. And I'm going to try to remember to tell you guys when it is, because they will all give a seminar on their work. And usually a job interview is basically guaranteed to be a great seminar, because there's a lot hinging on it. And so this is just you for your own curiosity, but I'll try to remember to mention them to you. Okay, so what are we going to be doing in the first section of the class? This is population and quantitative genetics, natural selection, genetic drift, et cetera, et cetera. Before I jump into the material, I want to just tell you in like 1 or 2 sentences what I do as a scientist. So, not surprisingly, I'm an evolutionary biologist. I work on this stunningly lovely plant, mostly, not completely. I mean, not all my work, but a lot of it is on this plant, American bellflower, or Campanula americana. And I do a lot of my research at Mountain Lake Biological Station, which, if you guys haven't heard of before, I'm going to talk to you about later in the semester and try to convince you that it would be a great thing for you to do this summer. They have summer classes up there, research opportunities, and basically students who go there, it's one of their PTBA experiences. So I'll talk about that later. And I do a lot of work out in natural populations. What kind of stuff do I do? Well, one of the reasons that I really love teaching this class is because basically what I teach about is what I do in my research. So I'm going to teach you about natural selection and genetic drift and inbreeding and heritability and plasticity and differentiation in populations and molecular evolution, and we do all of this stuff in my lab. So I am holding back and giving you not every example a Campanula example, so you can hear about things other than just this plant species. But I just tell you this because the kind of stuff that we're going to be talking about here, basically all the evolutionary biologists in the department work on this sort of thing. And so if you wind up being interested in anything that we talk about and you want to do research, you should consider contacting either me or other evolution people in the department. And the way that I'm going to talk about material, a lot of times what I'll do is I say, okay, this is what this concept means, and here's an example. And so hopefully when I give you the examples, part of that is so that you can have an understanding of how research operates in different areas. Okay, so that's the intro. Now we're on to business. And we're going to start off today with population genetics, talking about Hardy-Weinberg. Every lecture is going to have an outline on the front of the slide, whatever. If you're studying and you just kind of get lost, there's like all these slides and you're trying to figure out which are the points that really matter, how should I study, go to the outline. This tells you how I think the data is organized, what the big picture is, and if you understand the stuff on the outline, you probably understand the material that's covered in the lecture. So there's one other thing. So what you're about to see is, I have the kind of slides that don't have a lot of text on them. So what that means is that you're probably going to have to take some notes. If you probably have to take some notes, you'll probably have to come to class. So it's your decision, but I told you that I would never test you on material in the textbook that we don't do in class. On the other hand, I'm like totally comfortable testing you on material that we do in class that's not in the textbook. So my recommendation is come to class because you're going to do better. And hopefully you'll have more fun too. Oh, and there's one other thing. And Butch left. So about 3 days ago, Butch sent me an email with a link to a paper in it. And what the link said was it was a paper on learning, and it talked about using computers to take notes versus taking notes with your hands. Have you guys heard about this research? Yes, some of you are nodding yes. If you haven't, the take-home message is really simple. Students that take notes by hand get better grades than students that type on their computers. I have to admit that I was kind of taken aback by that. So the question is like, so what's the difference? You're here, you're doing something. And apparently, do you know?
Yeah.
All right, go for it.
Because you can't write physically as fast as you can type, your brain has to synthesize the information as you're taking it in, which means it sticks in your brain better.
Yeah, that's what this paper said. I thought that was really fascinating. So you basically have to summarize it when you handwrite notes, and when you type, you can just type along. Unless you don't type very well. But if you don't type very well, maybe you're not in such bad shape. Anyway, I just put that out there as food for thought. Your business what you do. Okay. All right, so getting into action here. So we're going to talk about evolution. When we talk about evolution, we need to think about genes, not surprisingly. And anybody in the classroom who's taken genetics has certainly thought a lot about genes. And the big thing about evolution is we need to think about genes in a population context. And so the question is, why? Why is it so important to think about genes in a population context? And that's because the definition of evolution is a change in the genetic composition of a population over time. Okay, so if you want to talk about the change in genetic composition, you got to be thinking about genes in the population context. Okay, so population genetics then is a study of these changes in genetic composition. A lot of time you're going to hear me talking about changes in allele frequency. That's just convenience. It's basically changes in any sort of genes. We could be talking about SNPs, single nucleotide polymorphism, alleles, larger chunks of DNA. And what we're going to do is use this first section on population genetics as a mechanism of reviewing the basic mechanisms of evolution. I'm pretty sure today what we're going to talk about, everybody in here has heard before. I'm kind of embarrassed to say that because, like, I want you to come and learn new things and get excited. But on the other hand, you might not have thought about evolution for a little while, so this is just a sort of one lecture to ramp everybody up and get you to the same point. Okay, so when we talk about changes in genetic composition, we need to first ask, where's all this new genetic variation come from? And the answer is mutation. Mutation is the source of novel, that is brand new genetic variation. And it's mutations that happen in gametes that are important because they get passed on to offspring. And what I'm going to do is review what are the three basic types of mutation. The first type of mutation is a point mutation. And the point mutation is when you have one nucleotide substituted for another nucleotide during the replication process, and this leads to a nucleotide change, or what I'll call a SNP, that is a single change in a single nucleotide polymorphism. We're going to talk a lot about SNPs when we talk about molecular evolution. Second type of mutation is a change in the arrangement of genetic material. What this figure shows here is an inversion, so we have a chromosome with just 5, or I guess it's 6 regions of the chromosome identified by letters, region A, B, C, D, E, and F. And what happens in inversion is you have breaks, and when they reanneal, the wrong pieces, so it should have reannealed like this, instead it's reannealing like this. And what that means is that this section is backwards or inverted relative to the original. Why is that important? Well, in evolutionary biology, we care about inversions because When you have an inversion, it reduces recombination because recombinant types are lethal. So basically, if you have an inversion in here, you wind up with, if you have an inversion and you have a crossover or recombination event within your inversion, you wind up with duplications and deletions, and so typically you don't get any recombinant gametes with it in this inversion. Sorry, that was a little long-winded. What I really want to say is, in terms of evolution, inversions and changes in arrangement of genetic material are important because they tend to lock together blocks of genes and reduce recombination. That's what I want to say. Okay. Translocation, fusion are other types of changes in the arrangement of genetic material. Third type of mutation is changes in the amount of genetic material. And this figure is from your textbook. I should say, always I will show down here where figures come from. Heron and Freeman is your textbook, so this tells you exactly where you can find this information in your text, so you can review it. So gene duplication is just saying you get a gene duplication when you get incorrect alignments, often in regions of a lot of repeats. And so when you get an incorrect alignment and you get a crossover event, you wind up with a duplication and a deletion. Missing here, 2 copies here. Okay, so we will talk about a lot about gene duplication and other changes in the amount of genetic material when we talk about how the genome is organized. So this is duplication of a gene. You can also have a duplication of the whole genome. This is called polyploidy. Brittany actually works on polyploidy and consequences of polyploidy for her dissertation. And what polyploidy is basically a doubling in the number of chromosome copies. So, for example, we are diploid. If we underwent a polyploid event, we would be tetraploid, 4 copies of each chromosome instead of 2. You don't find polyploidy much in animals, probably because when you change the number of copies of sex chromosomes, it messes up their ability to create the 2 sexes. Plants lacking sex chromosomes, you find a fair bit of polyploidy. So we will return to this again when we're talking about how the genome is organized. Okay, so that's just a quick run through of the types of mutation. The other thing that I want to mention about mutation is we often talk about random mutation. Mutation is actually not random. Some mutations are common, some mutations are rare. You actually have natural selection acting on the mutation rate to keep it low or to let it be higher. So in that sense, mutation is not random. Mutation, when we say mutation is random, we mean random with respect to the need of an organism. So if it would be useful to have a certain mutant, that doesn't make that mutant more likely to show up. So yeah, I think that's it on random mutation. Okay, so now I just quickly want to go through an example of how we think about evolution, leading us into Hardy Weinberg. So this example here is going to be about antibiotic resistance. So let's say you're a doctor. At this time of year, you treat a lot of patients that come in with strep throat. Say you're a pediatrician, you have a lot of kids coming in with strep throat. You give them all antibiotics, and some of the kids come back, they still have the symptoms of strep throat. So you're like, wonder what's going on here. I've given them the antibiotics, they're taking the antibiotics, kid still has strep throat. So you think to yourself, as any logical person would, the antibiotics must not be working. Why wouldn't they be working? They might not be working because there has been antibiotic resistance that has evolved within the strep bacteria. you can check that out, right? Because you could go and you could take these swabs from each of your patients. You have to do that to confirm their strep. You plate out the swabs on a petri dish. You get this little growth here, that's your strep bacteria. And then you can take little bits of filter paper that have been soaked in different sort of antibiotics and plop them on your different patients' petri dishes. And what you can see here from this example is some of the antibiotics kills the bacteria around it. We presume that's working. And some of them doesn't. We presume it's not working, right? So you could do this for all your patients. And in essence, you're trying to answer the question, do I have antibiotic resistance in my patients? And if so, what antibiotics are they resistant to? So really what you're doing is you're testing this hypothesis, right? That there's been the evolution of antibiotic resistance in your patient group. And anytime you ask a question like this, you have a hypothesis, whether you sort of formalize it or not, it's implicit that you also have a null hypothesis, right? Because you need to reject the null hypothesis in order to support the hypothesis you're testing. And so in this case, the null hypothesis is, no, the problem with my patients is not that they have evolved resistance to antibiotics. Maybe some of them aren't sleeping, and so even though they're taking antibiotics, their immune systems are so compromised they're not able to, I don't know, I'm making this up, right? But you know, maybe it's a different answer. So you always have to have the null hypothesis. Okay, so that's how you test for, evolution. Okay, so here comes the part where you get to see how much you remember from your previous classes. All right, to have the null hypothesis, that is, no changes in genetic composition, no changes in allele frequency, if that holds under, or that has 5 assumptions, or 5 conditions must hold for that to be true. So when you get no changes in allele frequency, just looking for volunteers, what has to be true? You can't have any, yeah.
Completely random mating.
You need to have random mating, okay? Yep. That's one. What else? Yeah. No mutation. No mutation. Okay? Yeah. No drift. Okay? We're up to 3 of them. What else? Yeah. No selection. One, 2, 3, 4. Yeah.
This one, isolated population, or is that?
Isolated population, that's great. Usually it's not put in that context. Usually what people say is no migration. So isolated population is absolutely perfect. Yeah, exactly. So these are the 5 things that have to hold to have no evolution. No mutation, migration, selection, drift, and random mating. If these things are true, if these conditions are met, you don't get any evolution. Okay. This is what's called the Hardy-Weinberg theorem, or we call these the Hardy-Weinberg assumptions, the Hardy-Weinberg conditions. You'll hear it referred to in a variety of ways. And period. Okay, I already said that. Here we go. And so now what I want to do is take you from this sort of conceptual level to a quantitative level. So the first part of the class, like the first material for the first exam, it's relatively quantitative, so people are going to say, should I bring a calculator to the exam? Yep. So you can expect to see some math. And what I want to do is work through examples of problems in class and then let you practice them in a review session and on your own. I forgot to say this in the beginning, but if you have a question, just raise your hand, stop me. Don't feel embarrassed because the class is big. Just do it. And especially if we're in this math stuff, that's when people are like, whoa, this is just like going a little faster than I can quite deal with. Just raise your hand. Okay, so Hardy-Weinberg, what I'm going to do is just give you sort of the most simple, straightforward way of thinking about Hardy-Weinberg quantitatively, and that is a one locus, two allele model. So the one locus will be the locus for A, where we have two alleles, big A and little a. And we're going to make three assumptions here: that individuals are diploid. That means they have two allele copies per individual, one from mom and one from dad. Oh, that's part of the sexually reproducing. And they have non-overlapping generations. There's all kinds of other configurations of Hardy-Weinberg that are more complicated. You don't have to have diploids, you don't have to have sexually reproducing, you can have overlapping, but they're just more complicated. And what I want to do is just go through with the most straightforward of these assumptions so that we can really focus on the evolution instead of hanging out with the math. Okay, so if this is true, if you have one locus and two alleles, basically you have three genotypes, right? Big A, the heterozygote, and little a. All right, because we're talking about a population, and our goal is to figure out changes in the frequency of alleles in the population, or how common they are, we need to give the alleles a frequency. So P is the frequency of big A in a population, in other words, how common is big A, and Q is the frequency of little a, how common it is. And when we look for evolution, we're just looking at changes in P and Q over time. Okay, and this is just a mechanism to help you check your math. Because there's only two alleles, big A and little a, you know that P plus Q are always going to add up to be 1. All right, so how do we get allele frequencies? Let's say we have a population of 100 individuals. 80 of them are big A. 10 of them are heterozygotes, and 10 of them are little a. In order to figure out P, the frequency of big A, we have to say how common is big A, right? That's pretty easy to figure out. In this population, you know that there are 170 big A's. How do we get that? Because each of these 80 big A individuals has 2 big A alleles, and each of these 10 heterozygotes has 1 big A allele. So there's 170 big A alleles in the whole population, right? There's 100 individuals, they're all diploid, so that's 200 total alleles, so the frequency of big A is just 170 over 200, or 0.85. Any questions about that? And then you can do exactly the same thing for little a. Or, you know that P plus Q is 1, so you know that little a must be 1 minus P. Frequency of little a. Okay, so this is starting from the number of individuals. P plus Q is 1. We can do the same thing starting from genotype frequencies. I showed you both of these mechanisms because I don't know which of these you've learned before, and I just want this to look familiar. So if we start with genotype frequencies, that is 80% of the population is big A, 10% is heterozygote, 10% is little a. To get the frequency of P, it is 0.8, because these are all big A, plus half of the frequency of the heterozygote, and so that's 0.85. Just a different way of doing exactly the same thing that we just did. So you can either start with the numbers, calculate it that way, or turn the numbers into phenotype frequencies and calculate it that way. Q, same deal. All right, now we get to the clicker situation. You know how to do this, right? So what I want you to do is take out your pencils and figure out what the frequency of P and Q is for this population. Because it's a clicker question, I got to give you some possible answers, and here they are. Okay? You know what you're doing? All right, let me turn it on. Some people are very speedy. Oh yeah, I don't have a totally welcome to talk to your neighbors and do clicker questions. Not a big deal. Yeah. Oh, do I use REEF? We used REEF last year, and so few people used it. We can enable REEF. Is there anybody else in here who would like to use REEF? A couple? Okay, can you email Brittany, and we'll just get REEF turned on? So if you don't have your clicker today, don't sweat it. Yeah. Where did I get the 200 from? Aha. All right, so where did I get the 200 from? So there's 60 of these guys, 30 of these guys, 10. So there's 100 individuals, right? And every individual has how many alleles? 2. Exactly. Yeah. Yeah. How are clicker questions are what? Into your grade? Good question, because I forgot to talk about it at the beginning. So, clicker questions, to be completely honest, it's mostly good effort for you. The way we use them when we calculate the grade is we go through at the end of the semester and we assign grades. And then we look at people that are near the grade distribution, and if they have high number of clicker responses, we will up them to another grade if they're close. So this is the thing, so since I'm a quantitative person, the honest truth is not that many people are affected, but you don't know whether you're going to be one of them or not. It's sort of the bottom line. Any other questions? All right, so you guys are done with this. Oh yeah, there's plenty of people who responded. So what I see is I don't get this. All right, let me, oh yeah, okay. What I see is you guys have it together. Practically everybody responded this one. P is 0.75, Q is 0.25. We're good with that, right? Anybody have any questions? Okay. Thank you. You know what you're doing. Oh, I have to get this turned off. When too many people answer the right question, it overloads the base station, and I can no longer understand what's happening. All right. So, another one. All right. Here we have no lilies, no homozygotes. Oh. Yeah, I know what you want. There we go. almost there? All right, what I can see is that no one has gotten this wrong. Everybody's gotten it. So the answer is, what's the answer? C, exactly. Okay, so which, if you might recall, is the same answer we got before, right? Now, I hate to say this, but we're just going to beat the dead horse here because I want you to do one more of these. So obviously you're doing this one now. Okay. They get faster each time, right? They get faster each time because you start realizing, let me look at this again here. The answer is this one yet again, right? So, let me just stop this. Okay. All right. So, yeah. So this is not only a change to practice, but there's a method to asking these questions repeatedly, and that is every single time you have the same allele frequencies, right? But the reason I wanted to do this is because what this shows you is you can get the same allele frequencies out of populations that have really different genotype frequencies, right? So you can always take genotype frequencies or the number of individuals in the population and calculate allele frequencies. Clearly, if you know the allele frequency, you don't necessarily know the number of individuals of the different genotypes in the population, unless that population is in Hardy-Weinberg. Because if all of these things hold, no mutation, migration, drift, selection, blah, 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 if all that holds, then you can take the genotype, the allele frequency, and calculate the genotype frequency. How do we do that? I think this is, again, something you've done before. If we know the frequency of the alleles are 0.75 for big A, 0.25 for little a, you can do a simulated random mating. I think of your good old Punnett square and say, okay, the frequency of big A is going to be 0.75 times 0.75, and the frequency of the heterozygote here, etc. You fill these all in, and you get from this frequency of big A under random mating with these other assumptions holding is 56%. The heterozygote, which is the sum of these two, is about 37%, and little a is about 6%, right? So you can take the allele frequencies, go through one generation of random mating, and get the genotype frequencies if Hardy-Weinberg holds. Later, we're going to make adjustments to these equations to allow for inbreeding, etc., where you'll find you can't do this. But under Hardy-Weinberg, you can do it. All right, so now what we can do is we can go back to, these are the three populations I gave you. So you might say, are any of these populations in Hardy-Weinberg? Well, remember, it would be like 56, 37, and 6 would be about the number of individuals in each of these phenotypes under Hardy-Weinberg. Obviously, none of them match perfectly, right? I didn't make them so they match perfectly because this gives you easier math to do. But this is probably the closest. If you really wanted to find out if the population was in Hardy-Weinberg, you would do a statistical test, like a chi-square test, to see if these numbers deviate from these numbers. But skipping the detail, if the number of individuals in the population is fairly close to Hardy-Weinberg expectations, that tells you that evolution is not happening, at least for that generation. Okay, so quick review. If you want to see the populations in Hardy-Weinberg, calculate the allele frequencies. You use those allele frequencies to calculate the genotype frequencies that you get under Hardy-Weinberg, and then you compare those to the actual number of individuals that you observe. Okay, so piece of cake there. We have like 4 minutes left. So I will briefly start something and then we'll get to it next time. So if I give you too many slides and we don't get to them, I'll just bring them back into the PowerPoint for the next class. We're going to start off by looking at natural selection. And this is what I thought we could just go ahead and quickly do this time. All of you guys have had before the tenets of these that have to hold for evolution to happen by natural selection. But quickly, there's 3 things that must be true for evolution by natural selection to happen. You need to have phenotypic variation in some trait, whatever trait, the phenotype has to vary. You need to have differences among individuals in fitness that are associated with the variance. Okay, so you've got different phenotypes. One of them has to yield better fitness than the other, and those variants have to be inherited. If these 3 things are true, evolution by natural selection happens. It's automatic. It just happens. And here I've just taken a slide. This is probably like the most text you'll ever see on a slide from me. A nice prosy way of saying how evolution works by natural selection. Evolution by natural selection means that individuals, you don't have to write this, it's on your slide, who have the form of a trait that enhances survival or reproduction will produce more descendants. And since that trait is passed on to their descendants, the frequency of that trait will increase in the population. Okay, so evolution by natural selection yields an increase in frequency of traits that are associated with high fitness. This is 10:48, and it's a very bad time to start an example. So I'm not going to, except to say, this is where we're going to pick up next class. See ya. If you have questions, just come on.
밥 먹으러 가요.